Implementing pharmacogenomics in clinical practice presents both challenges and opportunities. Here are some key factors to consider. Challenges. First, limited evidence. While there's growing evidence supporting the utility of pharmacogenomics in improving treatment outcomes, the evidence base for many drugs and genetic markers is still evolving. Limited availability of high quality clinical studies and guidelines can make it challenging to establish clear recommendations for pharmacogenomic testing and interpretation. Second, complex genetic variation. Human genetic variation is complex and diverse, with numerous genetic markers influencing drug response. Interpreting and integrating these variations into clinical decision making can be challenging. Additionally, genetic variations can vary among different ethnic populations, requiring consideration of population specific genetic markers. Third, cost and accessibility. Pharmacogenomic testing can be costly, and insurance coverage may not always be available or consistent. This can limit the accessibility of testing, particularly for patients with limited financial resources. Additionally, the availability of pharmacogenomic testing facilities and trained personnel may be limited in some healthcare settings. Fourth, lack of standardization. Standardization of pharmacogenomic testing methods, reporting formats, and interpretation guidelines is crucial for consistent and reliable results. However, there is still a lack of standardized approaches across different laboratories and healthcare systems, which can lead to variability in testing results and interpretation. Opportunities. First, personalized medicine. Pharmacogenomics allows for personalized medicine by tailoring treatment approaches based on individual genetic profiles. It offers the opportunity to improve treatment outcomes and reduce adverse events by selecting medications and dosages that are most likely to be effective and safe for each patient. Second, enhanced drug safety. Pharmacogenomic testing can identify patients at higher risk of adverse drug reactions, enabling proactive measures to minimize harm. By avoiding medications with a higher risk of adverse events or adjusting dosages based on genetic information, healthcare providers can enhance patient safety. Third, improve treatment efficacy. Understanding an individual's genetic variations can help predict their response to specific medications, enabling the selection of drugs that are more likely to be effective. This can improve treatment outcomes and reduce the need for trial and error in finding the right medication for each patient. Fourth, advances in drug development. Pharmacogenomics can guide drug development by identifying genetic targets, facilitating the design of more effective clinical trials, and aiding in the selection of patient subpopulations that are more likely to respond to a specific therapy. This can accelerate the development of targeted therapies and precision medicine approaches. Fifth, integration with electronic health records or EHRs. Integration of pharmacogenomic data with EHRs can provide clinicians with readily accessible genetic information to inform treatment decisions. This integration can facilitate decision support systems and assist in real-time interpretation of genetic test results, supporting personalized prescribing at the point of care. In summary, while challenges exist, the implementation of pharmacogenomics in clinical practice offers substantial opportunities to improve patient care through personalized medicine, enhanced drug safety, improved treatment efficacy, and advancements in drug development. Addressing the challenges will require ongoing research, standardization efforts, education for healthcare professionals, and collaborations among stakeholders to optimize the integration of pharmacogenomics into routine clinical practice.